So what a pleasure to have my friend Rinaldo here, and I'm going to bombard him for 15 minutes or so with interesting questions, hopefully. Rinaldo, so this meeting is organized by JAFRON, and it is supposed to talk about hemoadsorption and adsorption as a mechanism. What is the chance, in your perspective, that we can design a very good trial in adsorption?
I think at the moment it's low. I think what we need to do is to build up a body of evidence that creates a more specific population to intervene on. And I think we need to find ways of ensuring that we deliver the technique at the right dose. And, you know, I don't want to kind of injure the story of hemoperfusion by going too fast into randomized controlled trials without having an appropriate population, an appropriate dose, a full understanding of the absorptive capacities. I think there are some populations that offer a kind of an opportunity, a specific opportunity for investigation. I think we've got some specific groups of patients. I'm particularly interested in people with liver failure, people with acute liver failure, because we've already got very good data from you in vitro about bilirubin removal. It's very effective. We've got some in vitro stuff about biliary acids, which is a good thing. And I'm particularly interested in ammonia removal. And I think most people that work in the world of liver transplantation, like I do, are very conscious of the effect of ammonia on cerebral edema, and that's a real target for us. And so we are setting up. We've got ethics submissions to do pilot work in people with acute liver failure and see where that takes us. But again, it's all exploratory stuff.
So you mentioned today that we are today with hemoperfusion where we were 30 years ago with CRRT. At the time, there was no evidence for this, no evidence for that, and we had to build the entire story. What is the mistake that we made that you don't want to make again for this?
Yeah, it's interesting to look back, because you look back and you think about things that could have been done better, but you say that because we now know the end of the story, or at least chapter number 7 or 8 in the story, and it's very easy to be wise after the fact. We have a saying in Australia that there is a specific diagnostic tool called the retrospectoscope, and it should be used very sparingly. And I think the biggest challenge in terms of learning is that before we embark in trials, we actually need to do that kind of the grunt, slightly less exciting work about the biological effect, the physiological effect, the dose, like a lot of the stuff that is not that exciting. It's not like that powerful. Oh my gosh, we've done a randomized control trial of 3,000 people,
But this is a good message for young people, for example, that may not be able to design a large trial, but they can do a very specific study on adsorption. And they can help to add information to what we call to build the wall of evidence, right?
Yeah, I think
so it's a good idea, and everybody in the room should be encouraged to do maybe a small study. For example, we did bilirubin, we did vancomycin, we are doing linezolid. Maybe there is somebody else that wants to do one specific drug or one biological effect. Maybe in the lab they have information about HLA-DR or other things.
And and some of the things you were talking just now about the technology of combining hemofiltration or hemodialysis with hemoperfusion, with a core. There's a lot of stuff about the technology and the technique and the hemodynamics of the circuit and the pressures and all of that technical stuff that people kind of don't really want to worry about, but they are the necessary component to being able to deploy a therapy safely. And so, some of the stuff about citrate, well, what happens to ionized calcium, how did it work, and how was it played out, and all of that detail, people tend not to collect. The pressure, the resistance of the filter, the resistance of the cartridge, how does it change over time, and how does that predict potential coagulation and loss of function, all that kind of stuff. I think some of that can be done in vitro, but a lot of it has to be done in vivo. And so there are lots of opportunities out there for people that are undertaking this technology in the care of their patients to actually ask themselves, what can I collect that is useful to the world? Yeah.
Do you think that these studies may also leverage the utilization in other forms of blood purification? You mentioned liver failure, but also chronic kidney disease, for example.
Yeah, I think, again, you've been in the wearable artificial kidney for a long time, and I think the sorbents are still attractive, and they're still in a phase of evolution. And I think that's very interesting. And there's literature, certainly for the JAFRON cartridges in the treatment of pruritus in patients with kidney disease, and in people with liver disease as well. And so there are other potential indications that we are not discussing at the moment outside of the critical care environment, and they will provide also additional information. And I think doing some sorbent work within the chronic dialysis environment would also provide some really good information that can be then transferred to the critical care environment. So there's a lot of stuff that needs to be done. Yeah.
So looking at the program of this meeting, where we try to find the most recent data, etc., do you think we are in a phase where we are moving forward slowly, but we are moving forward, or are we stuck in some...
No, I think we're moving forward. I think, we got programs underway. Tomas here is just preparing to do some work in burns patients. John Prowell had to leave, but, he's putting in ethics submission to look at trauma patients. You're doing some really good work in vitro. We're setting ourselves up to do lots of experiments in sheep, to do some of this kind of basic work about what happens to biology and drugs and toxins. And so I think, I think even a year from now, maybe a year and a half from now, but we will have a lot more data and a lot more systematic.
After this meeting, we will have a fantastic chance because we will do an ADKI meeting on sepsis.
That's true.
So the good thing about ADKI has always been that it pushed forward the envelope and trying to stimulate new research and moving more from the unanswered question that rather the certainties. So for people who are passionate about guidelines, we should recommend that they should not be depressed or frustrated, because guidelines can only tell you that the sky is blue because everybody consent on this. But before you give a guideline, most of them, by the way, are opinion-based. We should say that.
That's right.
ADKI is probably more, has more humility, has more, let's say, modesty in trying to recommend studies to understand better things. So we will have a fantastic chance in the next days.
Look, I agree. I think there is much to be done that is useful in indicating some of the directions that we need to go and helping people understand what they can do to contribute to knowledge. There is so much that we don't know and so little that we know that trying to systematize and do it progressively is going to be a good thing. Yeah.
Looking at the spectrum of sorbent that JAFRON has,
Yeah.
it's my impression that there is there a treasure that we have to discover.
I know, because, the other bit, of course, this has been true of blood purification therapy from the beginning. There's an interaction between industry and clinicians. And so industry provides technology and they advance the technology. The clinicians look, okay, that looks really interesting. They start using the technology and they say, well, that's great. It does this and it does that, but maybe we can do that the other. And then you go back to the technology and it's, we can modify the beads, we can modify the pores, we can modify the coating, we can modify the size of the cartridge, we can modify the flow profile. And it's a continuing interaction to optimize the therapy. And I think that's going to happen, I think, in the next 5 years. And it's going to be good for industry and it's going to be good for clinicians and patients, of course. Yeah, I think that's absolutely right. Yeah.
So that's fantastic. Now, in the room, we have time for one question. Is there anybody who wants to ask a question to Professor Bellomo? You have a unique chance, my friends. Otherwise, I'm forced to do. Thomas, you may have a question.
Come on, Thomas, speak up. Very serious, okay, all right. You can shout. Very polite.
So, Rinaldo, we don't have so much data on how to use in clinical practice this device for the length, the blood flow, and the number of sessions. I know we need to do all the works, but what do you recommend from now without all this data?
Looking at the some of the in vitro work, obviously we have the cartridges that are available. And I think a blood flow around 200 mils a minute is very reasonable to begin with. Maybe we can go a little bit higher, but no one's actually done the relationship between extraction ratio and flow. So at what point the flow is too fast and how can you optimize? There'll be in vitro work. I'm sure Claudio will do it. I'm sure he's onto it. But that's important. So at this moment, you'll be operating about 200 mils a minute of blood flow. And of course, the data that we have in vitro is that, most of the absorption is played out by about 3 hours. And then after that, there's an exponential decay of that. And that highlights the fact that you got to have some target molecules because, if you get to about 3 hours or so and then there's exponential decay in your ability to remove some target molecule, you might yet be able to hold the level steady and still come down because you might have affected the production. So we need to have some target molecules. That's why I'm interested in the liver patients because we got potentially 3 target molecules, bilirubin, bile acids, and ammonia. In burns, for example, you might have some markers of inflammation, and that would be what happens. And if you're able to, for example, use interleukin-6 as a broad marker of inflammation or ferritin or other markers, and you're able to hold them down, that might say, okay, we can stretch out the procedure for a bit longer. But at the moment, I'm thinking 3 hours, you probably should change the cartridge.
Well, we have seen that there is a kinetics, for example, of bilirubin, that in vivo reminds very much what is occurring in vitro. So we may know exactly. It is clear, if you use 300 ml per minute of blood flow, you will have an extraction ratio which is much lower compared to what you have if you use 100 ml per minute. But then a problem of blood viscosity, clotting in the device, flow distribution may enter. So the 200 ml per minute, also for me, it's a very good compromise. And we have done studies on what is defined as mass transfer zone, which is how the sorbent gets saturated from the entrance of the cartridge progressively, and it is probably moving up to 6 hours if you use the large sorbents. For this reason, we use mini modules, and with this we can identify maximum adsorption per unit or per gram of sorbent.
That's right, yeah.
So that defines the maximum capacity. And this maximum capacity is interesting because you can also study if you can desorb substances,
Yeah, that's right.
which seems not to be the case because hydrophobic bonds are very tight. And so are going to study all these aspects. We did the CT scan studies on flow distribution.
Looks good.
And it's very interesting. So all these pieces may help to add to the story. One
it's
last
like one of
question.
those puzzles, and you're just putting the pieces together until you start seeing stuff.
One last question because it's very late. Are you excited about adsorption?
I feel a little bit young again.
Exactly.
It brings back memories of the late 1980s when we were doing this kind of stuff about continuous renal replacement therapy. And you know, those days we were putting arterial cannulae and venous cannulae and letting the blood pressure drive the flow into the filters and moving the filter up and down to change the drip rate in order to increase ultrafiltration. And at that time, when we were doing continuous arteriovenous hemofiltration, and then we had to go to the peritoneal dialysis nurses and steal peritoneal dialysis fluid to let it run counter current using volumetric pumps that we now use to give intravenous fluids in order to add dialytic. Like, people thought we were crazy, and maybe we were, but you know, it looked like we will never get anywhere. Now, we just finished a session where you're actually talking about machines integrating ECOR, hemoperfusion, hemodialysis, automated with pressure controls. It's another planet. So I'm feeling a little bit younger again.
I have to tell you this story. In the mid-'80s, I went to visit Rinaldo in Pittsburgh when he was in Pittsburgh. And at that time, we had hemofilters with one port for the ultrafiltration, so we could only do hemofiltration. And I said, Rinaldo, why don't we transform this into a dialyzer and we do a countercurrent flow like hemodiafiltration? He said, how would you do that? Well,
Make a hole.
Came back, I made with the drill a hole in the unit, and then we fixed with the glue and then re-sterilized it. Today they will.
We're not going to do that with hemoperfusion. The technology is better. It's okay. It's okay. Claudio's not going to put the beads in with his hands. It's okay.
But it is interesting that all these things eventually transform into reality. So I also feel younger again.
There you
so
go.
Rinaldo, we will do nice things. Thank you very much for all, and thanks for staying until this late time.